is actually Freddy's, because Friederike Erhard, is leading all this work on Rett from my group. And she actually made most of the slides. Rett is a nasty disease. It's one of these rare diseases that is monogenic, and it causes nasty effects, growth disorder, both motoric and in communication skills at a low age for children. And it's this typical thing that you see only now and then because it's one gene being highly affected or highly affecting a lot of things. It's really nasty to see that. And of course the problem is because, and that's the typical rare disease problem, because it happens not often, it's really hard to get enough data to even study it. So what we know is that there is a single gene involved in that, and which gene that is, and that is MACP2. And that is an epigenetic core-regulating gene that affects a lot of other things, basically through gene expression, but also affecting the chromosomal structures and things like that. It basically, the first function is actually triggered by binding to methylated DNA. So one of the first things we did is we created a wiki pathways pathway for that stuff to just describe what we know about MACP2, basically using the pathway as a kind of literature review. That also led to the interesting understanding that that gene that now turns out to be a GOR epigenetics regulator actually was not present in any of the other pathways, which shows you how important rare diseases can be to really understand even core processes, because we actually only understand them when they break. All right. Now, once we have that, we actually publish that thing, it starts to become interesting to map data to that, right? You want to know about these genes in this pathway and be able to map. And we have a mapping tool to that, which is actually one of the core ELIXIR mapping tools, because it does identifier mapping in general, and it can do that in this tool, but like you see in a lot of other things too. And you can integrate it thus in R pipelines or cytoscape workflows and things like that, right? All using the same kind of mapping data and the same mapping tool basis. And if you do that, it allows you to do things like that. Now you can browse that pathway, and you can just click that MACP2 genes, and you collect all these links out, so it starts to look like Biomart. It's actually based on Biomart, of course, but it brings Biomart mappings into all kinds of tools, making them more interoperable. Right? And then we start, and I missed that one, we actually got a lot of data that was available in existing data repositories. Of course, that links to, could we actually find that? Not so good yet, right? So the whole repository annotation is there. It follows Miami standards and such, but looking for Rett data, Rett-related studies annotated in such a way that you really find them if they're not from patients, but from animal models that are used for that, turned out to not be trivial. So there's still a lot of work to do there on findability in these resources. But it also shows how important that is, because you really want to connect that kind of stuff to a disease like that. They do these studies and then they're basically hidden, even in the repositories. Right? We published that one too. Then from there on, you can move on, right? You can take the pathway, convert the pathway in a network, and that doesn't make it any better understandable for most of you, but it allows you to start connecting things using all kinds of interoperability approaches that we have for these connections again. Right? Like, we can extend that network, adding the drugs to it, and see where in that whole process druggability is currently a possibility. Like where Marco often says, hey, if I see the same phenotype, I might look at a drug that I could use for these same phenotypes. You also need to know whether the drug actually hits the process you're looking at. And this allows you to do that. Right? And same thing for regulations. Right? As soon as you have that pathway, you can extend it with transcription factor interactions, providing you have a resource available that really knows about what the transcription factor targeting things are, which is basically a mapping approach from your transcription factor to your targets again. Right? Now, the question that comes up is then, is it a genetic disease? So I'm basically pestering you with all kinds of other omics data. What about the genetics data? And can we use that to understand why these phenotypes for different patients are still different? right? Maybe there is something in that genetics that says if it's this part of the gene, you get another effect than if it's that part. Was that even studied? No, rare disease, little data, no possibility to do that anywhere. Can we get enough data and start to combine that, right? So this is the question I just said, like, here are the possible variations. Can we connect that to something like these pathways again, right? Well, you can. So basically we created, as part of an ELIXIR implementation study, a new BridgeDB database that maps variants to genes for any use case, and used it here. And you see the simple explanation here. If you look at the right, you now see all the SNPs occurring for that specific MACP2 gene. And in principle, if you would click any other gene in any other pathway, you would get the same information. And you're now able to connect genetic variations to the genes, to the pathways in different tools that use that same BridgeDB mapping already. All right? And then they said, and now we have a lot of groups in this community, and they're basically all doctors that saw more than one patient, right? And then started to collect all the patients around them from other practices, and said, we built this database, which typically is a spreadsheet, with information about that disease, and I thought that this was the best way to do it. And they all have the best way to do it, and they're all right. The problem is, you don't get them connected too easy. And that is what this ELIXIR Bring Your Own Data sessions are for, right? You bring people that have that type of data together with data experts. Probably all the faces you recognize are the data experts, the others are the red experts, right? And a few people from my group that just joined to learn all these things. And then you can start to work on getting that information in a linkable way and start to ask the question, can we now understand when SNPs are in a specific region, because now we made that region specific for all the different data, actually do something in common. Now that is where we are, because Freddie actually planned a meeting to do exactly that next Monday. So we basically have in mind then that these different variants will actually then not just map to each other, but possibly map to the different outcomes of the disease, or not. But that is a real research question. This is my final slide then. It's just indicating that what I told you about is not just about Rett, it's also about ELIXIR. So yes, we use a lot of tools, and all these tools just pop up and go by on my slides, but they are all described in the tools platform, and that is where you can use them from. It's Rett data. We're basing a Rett database platform. We're also evaluating all these databases for genetic variations that have these smaller sets, which is directly linked to the human data platform. Of course, I even talk here from interoperability. We're setting up tools training for the rare disease community, and that's then the fourth platform. It's really the rare disease use case. I showed you a bring your own data session, and in fact, most of this work was done in 2 implementation studies. And from that, I can take some questions, or we can take some questions.
So I have a question. So my understanding of what you presented was that you built a platform to integrate and explore the network of what's known around some of these rare diseases. In one of the rare disease situations that I worked at, or we've been working on, there was a little bit of a different scenarios. Like, I have a rare disease, and yeah, I have a knowledge network that I know around that rare disease, but I also have this, a gene or a metabolite. The mechanism, the relationship is unknown, right? It's observed phenotypically in a model organism or something like that. And the question is, what are the possible mechanisms that join these two things? Does your infrastructure address that use case at all?
Well, we stumbled on the same questions. That was actually the red lines in the one that showed green and red connections from that MACP2 gene. Does it really allow that? I'm not going to answer that in 10 seconds. I have to think about that.
Yeah, my 2 cents would be that it can be either in both. So maybe you can distinguish as there's the enabling part, which is sort of fitting in building infrastructure. And there are specific cases to work on, like Rett syndrome right now, where you go deeper, and at BY, these bring your own data workshops, you can also go deeper into a case. So I think it's both, we're trying to do generic things that then people can implement, and yeah, and obviously there we have to make choices. We can do a few cases, but not all of them from an ELIXIR point of view.
I guess the interesting technological thing is that when you open yourself up to the second use case, you in theory have to be able to access the entire knowledge network from any different source, which becomes a challenge, but also
and that's the enabling part. Yeah.
Thanks.
Yeah. One quick question on that point. So how do you choose which bring your own data to organize and to choose the themes then? That's a good question. So one is it's driven by the need from the rare disease community that we work with. So a lot, for instance, is working with registries, and this summer school, the Health Institute in Rome organizes that summer school, and they have a big say in what who is being invited. It's always oversubscribed. And for this rare disease data linkage plan, there's an element of it's funded by different projects, but there's also an element where stakeholders have to put in their own investment. So that's can be in kind, just because they provide their expertise, but they have to do that. We require that, and that's how we prioritize rare diseases. It's just those that are willing to do that, those will be working with. It's a way of doing it.
I think it's really important. We have done a number of these things now, and we see typically you get different different domain experts every time, because we really do it for a different domain every time, but the pool of data experts available to do that is way too small. So these things really have to become more train-the-trainer sessions in part to just extend that pool. That is something we really have to work on now.